Hi, my name's Joyce Wang. I'm a postdoctoral researcher at the University of Michigan in the Department of Microbiology and Immunology. Today, I'm going to tell you a bit about how we study the transmission of drug-resistant bacteria in nursing facilities. Healthcare-associated infections affect many people in this country and pose a major financial burden to the system. Many of these infections are caused by multi-drug resistant organisms, or MDROs. Our group is deeply interested in understanding how these MDROs spread in the system. It is critical in helping us identify more effective intervention strategies. One of the main reasons why it is so difficult to control MDROs is because the healthcare system is in fact a very busy network. Hospitals, clinics, home care, and nursing facilities can share the same patients that may be carrying MDROs that travel with them to different facilities. So this cartoon illustrates how MDROs, in this case, a very resistant group of germs collectively known as CRE, can go from one facility to another. So in this scenario, this lady Jen, she has a stroke and is in the hospital. She is stable, but needs long-term critical care at another facility. So now Jane gets sent to another facility where other patients in this facility have CRE. A nurse doesn't wash his hands, and CRE are spread to Jane. She develops a fever and is put on antibiotics without proper testing. So now Jane becomes unstable and goes back to the hospital, but her new doctors don't know she has CRE. A doctor doesn't wash her hands after treating Jen. CRE is now spread to other patients. So the consequences of catching CRE can be very serious, as CRE can cause serious infections in the urinary tract, lungs, blood, and wounds. So it is really in everyone's best interest to prevent this from happening. The moral of the story is that if healthcare facilities continue to implement infection control on their own, independently without communicating with each other, more patients will get infections because patients go from one facility to another. To understand how MDROs can spread in Michigan, we collaborate closely with Dr. Lona Modi to collect MDRO samples from nursing facility patients, study them in the lab, and ultimately, we hope to use this information to improve patient care by intervening with MDRO transmissions. In a recent study conducted by Dr. Modi's team, 6 nursing facilities were recruited in an active surveillance program. Most of the patients were newly admitted from discharged acute care hospitals. During the study, Dr. Modi's team collected culture samples at multiple body sites from hundreds of patients at baseline, which is enrollment, 2 weeks, then monthly up to half a year, and looked at the colonization pattern at admission, which is again enrollment. And as you can see, many of these patients already came in colonized. For example, 44% of the patients were colonized in their rectal area at enrollment. So here I will break down the dynamics of MDRO colonization a bit further to show what is happening in the nursing facility. So almost 60% of the patients were colonized with or asymptomatically carrying at least one MDRO at admission, which is shown as the yellow circle here. And this number looks static over time if you look at the overall colonization pattern at the facility level. However, there's more nuance to this observation. During their stay, some patients lost their MDRO, but some gained new MDROs, indicated by the blue and the red arrows shown in this figure. So this sends a very clear message that while some patients were able to clear their MDROs, some became exposed to opportunities for MDROs to colonize their body sites. And this really highlights the critical role that nursing facilities play in influencing the clinical outcomes of their patients. And what is really interesting is that this high colonization rate was not driven by just one type of MDRO. In this bar graph, we're showing the 6 facilities enrolled in this study, each as a bar, and the height of each bar indicates the total number of patients colonized with different kinds of MDROs, such as MRSA and VRE. Each color in these bars indicates a different MDRO type. What's striking is that we found similar MDRO types such as MRSA, VRE, and E. coli in every facility. So now the main question that we wanted to ask is whether these facilities have acquired their MDROs independently or the same strains were circulating in all these facilities in the region. So to answer this question, we analyzed the MDROs in the lab. We used a high-resolution typing technique to better understand the relationship of different isolates. The technique is based on genetic similarity. For example, if I'm colonized with MRSA in my nose and I sneeze on my colleague Karen and I will likely carry genetically very similar or virtually identical MRSA strains, and we can use this information to infer person-to-person transmission. On the other hand, if Bonnie, another colleague, she has acquired a MRSA strain during her trip to New York, her MRSA will be likely quite different from what Karen and I carry, and her isolate will be further away from our cluster. So similarly, we can apply the same concept to isolates we collected from different nursing facilities. In this example, we have 6 isolates, 5 collected from facility A and 1 collected from facility B. The majority of the facility A isolates cluster together, so that suggests person-to-person transmission within facility A. But this facility A isolate over here clusters together with an isolate from facility B, suggesting the isolates from these facilities have mingled at some point and provides evidence for transmission between facilities. And as a reminder, both facility A and B are nursing facilities where we don't really see a lot of direct transfers between nursing facilities. So also important to think about where these patients are discharged from and readmitted to during their nursing home stay, because common healthcare exposure is also a risk factor for transmission. So to wrap up this talk, the take-home message is simply that MDROs are common in nursing facilities. Results of our study show that teamwork is critical, whether it's different disciplines within a nursing facility, such as nursing and environmental services, or between different types of healthcare settings, such as long-term and acute care facilities. Environmental cleaning and hand hygiene are basic, but crucial steps in preventing healthcare-associated infections. So in this slide on the bottom, we included photos of an educational activity at a nursing facility. The nursing and environmental services leaders applied a powder on room surfaces prior to room cleaning. Under an UV light, the areas before and after cleaning were photographed and shared with staff for teaching purposes. Forms, tools, and checklists on cleaning and disinfection can be accessed under the form section on the PRISM website. And last but not least, we would like to thank all the PRISM facilities for their time and all the hard work. We would like to thank the CDC and NIH for the funding opportunities.